Welcome to the deep dive. Today we're tackling metastasis, the process by which cancer spreads. Understanding this is, well, it's absolutely crucial because, as our sources show, it's this spread, not the original tumor itself, that's behind most cancer deaths.
That's exactly right. We've looked at a really detailed review article on metastasis from PMC and also information from the National Cancer Institute, the NCI, and meta-knowledge.
Okay.
And our goal here is really to pull out the key insights. How does it work? What are the big challenges? And importantly, what new therapies are emerging. Think of it as a quick but thorough way to get up to speed.
Okay, good. So let's start right at the beginning. What is metastatic cancer exactly?
So metastatic cancer is basically when cancer cells have left where they started, the original tumor, and set up shop somewhere else in the body, somewhere distant.
Right.
For a lot of cancers, this means it's stage 4. And what's really interesting is that these cells, even though they've moved, they still keep the characteristics of the original cancer.
So if, say, breast cancer spreads to the lung, it's still treated as breast cancer.
Exactly. It's called metastatic breast cancer. And yeah, it's treated as stage 4 breast cancer.
Yeah.
Being able to trace it back is vital for treatment decisions.
Makes sense.
Although sometimes, and our sources mention this, it's called cancer of unknown primary, or CUP.
CUP.
Yeah, that's when doctors just can't pinpoint where the cancer actually originated, even after looking really hard.
Right, when the starting point is a mystery. Okay, so how does the spread actually happen? I mean, cells don't just pack up and leave, do they?
No, not at all. It's actually a really complex multi-step journey. It's called the metastatic cascade.
Cascade.
Think of it like a series of obstacles the cancer cells have to overcome.
Yeah.
First, they have to invade the normal tissue right next to them.
Get out of the immediate neighborhood.
Right. Second, they need to get inside blood vessels or lymph vessels. That's called intravisation.
Breaking in.
Third, they actually travel through the bloodstream or the lymphatic system, catching a ride, essentially.
Like getting on the highway. So what happens when they arrive somewhere new?
Well, the fourth step is stopping, usually in tiny blood vessels far away.
Yeah.
Then they had to get out of those vessels, that's extravasation, and into the tissue around them.
Okay, so invade, get in, travel, get out.
Yes. And fifth, if the new place is, suitable, they start to multiply. They form a tiny little tumor, often too small to see on scans. That's a micrometastasis.
Micrometastasis. So it's there, but kind of hidden.
Exactly. Flying under the radar. And then the sixth step, and this is crucial for it to become a real problem, is angiogenesis.
Angiogenesis. New blood vessels.
Precisely. They need to trick the body into growing new blood vessels to feed this growing cluster of cells. Once it gets bigger and has its own blood supply, we call it a macrometastasis. That's the detectable tumor.
Wow, that sounds like an incredibly difficult series of steps for one cell to manage. What are the odds? Like, what's the success rate?
That's a really important point the sources emphasize. The vast majority of cancer cells that break away from the main tumor actually die somewhere along this journey.
Really? Why?
Well, they face all sorts of challenges. Physical stress in the bloodstream, attacks from the immune system, and simply not having the right signals or environment to survive in these new places. It's a real uphill battle for them.
So only a tiny fraction actually succeed. And I remember reading something about dormancy. Some cells can survive, but just wait.
Yes, dormancy is another fascinating and frankly challenging part of this. Metastatic cells can arrive at a distant site and just stay inactive, quiescent.
For how long?
For months, sometimes even years. And then, for reasons we're still figuring out, they can suddenly wake up and start growing again. This delayed recurrence is a huge issue in cancer care.
It really complicates things. Okay, so this whole cascade, the PMC review, breaks it down into 3 main phases, right? Dissemination, dormancy, and colonization.
That's right. Dissemination basically covers those initial steps we talked about: invading locally, getting into the vessels, intravasation, surviving the journey as circulating tumor cells, or CTCs.
CTCs, okay.
Then getting out of the vessels, extravasation, and finally becoming disseminated tumor cells, or DTCs, just sitting in the new tissue.
And these CTCs, the ones traveling, they don't always travel alone, do they? I read something about clusters.
That's a really interesting finding. Yeah, CTCs can be single cells, but also they travel in small groups, microclusters. And sometimes these clusters even include other cells, like platelets or immune cells.
Why would they do that?
It seems to offer protection, like a little shield, during the tough journey in the bloodstream. And maybe it even helps them when they arrive, boosting their ability to start a new colony, traveling with an entourage, basically.
Strength in numbers. That's quite a strategy. Okay, so after dissemination, if they don't immediately start growing, they enter this dormancy phase.
Exactly. Dormancy is when these DTCs are there, but they're not really dividing or forming a detectable tumor. Clinically, you wouldn't see anything on a scan.
So what are they doing? Just sleeping?
Well, the review suggests a couple of possibilities. They might actually exit the cell cycle, go into a sort of paused state, or it could be more dynamic. Maybe they're trying to grow a little, but the immune system or the local environment keeps them in check, suppressing any expansion. A kind of stalemate.
Okay, so they're present but controlled or just inactive. What eventually pushes them out of dormancy?
That leads to the third phase, colonization. This is when the dormant cells finally manage to adapt properly to the new environment. They essentially figure out how to work with or co-opt the local tissue, the tumor microenvironment.
They become metastasis-initiating cells, MICs.
That's the term, yes, MICs. They break out of that dormant state, start dividing effectively, and eventually form those macrometastases that cause organ damage and symptoms.
Right, so it's this whole journey. Leave home, survive travel, maybe hide out for a bit, and then successfully set up a new growing colony. It makes sense that cancer cells would need special traits to pull all this off. The review mentions genetic selection versus plasticity. How did those differ?
So genetic selection is the idea that within the primary tumor, which is usually a mix of different cells, some cells might just happen to have specific genetic mutations.
Right.
And these mutations give them an edge, maybe helping them survive the journey or grow better in a new place. This selection could happen late, or maybe even early on. Cells could spread early and then evolve separately.
And the local environment, the TME, plays a big part in which cells get selected, right? It's not just the cell's own genes.
Absolutely. The TME, all those immune cells, supporting stromal cells, the matrix holding things together, it's constantly interacting with the cancer cells. It creates pressures.
How do we know this?
Well, newer techniques like spatial genomics let us see where different cells are within the tumor. And we can see patterns like certain cancer cell types clustering with fewer immune cells, suggesting they're evading attack.
So they can evolve to hide better.
Exactly. They might lose markers on their surface that the immune system recognizes, for instance, things called HLA alleles. It's active selection for immune evasion.
Are there specific big genetic changes like copy number alterations that are common in metastases?
Sometimes. Metastases can show more changes like duplications or deletions of big chunks of DNA compared to the primary tumor. Amplifying certain genes, like MINFIC, has been linked to metastasis, potentially by changing the immune environment. But, and this is key, the review stresses that genetic changes alone often aren't enough to explain why a cell becomes metastatic.
Which leads us to plasticity. If it's not always in the genes, what's plasticity about?
Phenotypic plasticity is basically the cell's ability to change its behavior or characteristics without changing its underlying DNA code. It's about being adaptable.
So changing how it acts, not what it is fundamentally.
Kind of. It allows cells to dynamically shift in response to stress, like the harsh conditions during travel or the different environment in a new organ. They can switch between different states, maybe become more stem cell-like or more resistant to certain drugs.
They can change their identity on the fly, almost.
In a way, yes. Studies, especially in animal models, show cells going through multiple changes as they metastasize. And it seems some primary tumor cells might just be inherently more plastic to begin with, making them better candidates for spreading.
That's interesting. And a molecule might do one thing in the primary tumor and something totally different during metastasis.
Yeah, the context matters hugely. A gene that promotes growth at home might be involved in survival during travel.
So how do they make these plastic changes? What's going on inside the cell?
Several mechanisms are involved. Epigenetics plays a huge role.
Epigenetics changes on top of the genes.
Exactly. Things like DNA methylation or histone modifications. They can quickly turn genes on or off in response to signals without altering the DNA sequence itself. Then there are key proteins, transcription factors that control whole networks of genes involved in plasticity, and even things happening after the gene is read out, like how RNA is processed or modified, allow for really rapid adaptation.
It's like having multiple levels of control they can tweak. And what about energy? Metabolism must be critical too.
Oh, absolutely crucial. Metabolic flexibility is key. Cells in transit might face low oxygen or scarce nutrients. They need to adapt.
Huh.
Often, they increase their use of fats for energy. They might use processes like autophagy, basically recycling their own components to survive when starved. They have to switch between building things up, anabolism, and breaking things down, catabolism, depending on the stage they're in and the resources available.
It's just amazing adaptability. Now, another fascinating idea from the review is the co-option of developmental and regenerative programs. What's that about?
This suggests that cancer cells don't always invent new tricks. Often they reactivate powerful genetic programs that are normally only used during embryonic development or when tissues need to repair themselves after injury.
So they're hijacking the body's own building and repair toolkits.
Pretty much, yes.
Yeah.
Think about normal tissue stem cells. They have built-in abilities to divide and differentiate. Cancer stem cells seem to tap into similar programs. Or during lung repair, cells might temporarily become more primitive to rebuild tissue. Lung cancer cells might reactivate those same programs to help them spread.
An EMT, epithelial mesenchymal transition, fits in here, right? That's a developmental process.
It's a classic example. EMT is how epithelial cells, which are normally stationary and stuck together, can become more like mesenchymal cells, migratory and invasive. This is essential for development, like forming organs, but cancer co-opts it for invasion.
And it's not always a full switch. You mentioned hybrid states.
Right. Increasingly, research suggests that cells don't always go full EMT. They might exist in a partial or hybrid state, keeping some epithelial features while gaining some mesenchymal ones. This hybrid state seems particularly effective for metastasis. Maybe they get the best of both worlds.
Mobility and some ability to stick together or grow.
Perhaps. And once they arrive somewhere new, they have different ways to colonize. The review mentions elasticity basically recreating the original tumor structure, deformability staying in a more adaptable, maybe stem-like state, or even transdifferentiation changing into a completely different cell type suited for the new organ.
Incredible flexibility. But then there's the immune system. Our bodies should fight this off. How do metastatic cells get around that?
Immune evasion is critical for metastasis to succeed, and it seems to be a progressive process. The tumor and its environment become more skilled at suppressing the immune response over time.
What's the evidence for that?
Well, one stark example comes from organ transplants. Sometimes a patient receiving an organ from a donor who had an undetected cancer develops metastases from that cancer, likely because the recipient is on immunosuppressant drugs.
Wow. So suppressing the immune system allows hidden cancer to spread.
It highlights how important immune surveillance normally is.
Yeah.
And of course, the success of immunotherapy, those immune checkpoint inhibitors or ICIs.
Right, drugs that take the brakes off the immune system.
Exactly. They show that boosting the immune response can fight cancer. But importantly, the sources note that ICIs often work better against primary tumors or smaller metastases than against large, established ones.
Why is that?
Larger metastases have often developed more complex ways to suppress immunity locally. Some are cold tumors, they just don't have many T cells inside them to be activated. And even if they respond initially, the cancer can evolve resistance, perhaps by exhausting the T cells.
So the metastatic tumor actively creates an immunosuppressive bubble around itself.
That's a good way to put it. As DTCs arrive, they face local immune cells like macrophages and NK cells. They evolve ways to hide from them or shut them down, reducing antigen display, increasing inhibitory signals, releasing suppressive factors. The whole microenvironment, immune cells, stromal cells, the matrix gets remodeled to become more tumor-friendly.
And this isn't just local. Primary tumors can prepare distant sites before cells even arrive, these pre-metastatic niches.
Yes, that's a key concept. The primary tumor can send out signals molecules released into the blood that travel to distant organs and start changing the environment there, reprogramming immune cells, making the soil more fertile for when the cancer seeds arrive.
Even lymph nodes, which we think of as filters, can actually help the cancer spread systemically.
It seems so. They're not just passive stopping points. When tumor cells colonize lymph nodes, it can trigger broader immune suppression throughout the body. For example, interactions in the lymph node can lead to tumor cells expressing more PD-L1, which is an immune checkpoint, and can promote the generation of regulatory T cells that dampen immune responses elsewhere.
So spreading to the lymph nodes can actually pave the way for spreading further by weakening the body's overall defenses. Okay, let's circle back to dormancy again. It feels like a really critical bottleneck.
It absolutely is. The fact that cells can survive undetected for potentially years and then cause a relapse is a massive clinical problem. And how long they stay dormant varies a lot between cancer types.
What keeps them dormant? Is it just lack of signals to grow?
It's likely a combination of factors. Cellular plasticity is key. Dormant cells often adopt different characteristics, maybe more mesenchymal traits. Specific molecules seem to help maintain dormancy. Epigenetics is involved. silencing growth programs. And there are inhibitory signals coming from the surrounding tissue, or even from the dormant cells themselves, putting the brakes on proliferation. Plus, adapting to low oxygen or nutrients likely forces them into a slow-cycling or non-cycling state.
Can they also hide from the immune system while dormant?
Yes, that seems to be part of it too. They might downregulate things that NK cells recognize, for example, making them less visible to immune attack while they're quiescent.
So what wakes them up? If they're dormant for years, what changes?
Several things can trigger reawakening. Inflammation seems to be a major factor, maybe from surgery, infection, even smoking. And importantly, the process of aging itself seems to play a role.
How does aging affect dormant cells?
As we age, tissues change. The bone marrow environment shifts. The extracellular matrix remodels. There could be more background inflammation and maybe a decrease in the factors that normally help keep cells dormant. These age-related changes in the host soil might provide the right conditions for dormant seeds to sprout.
That's sobering, that the body's own aging could contribute to cancer relapse. Okay, shifting gears slightly, metastatic tropism. The idea that cancers prefer certain organs. Why does breast cancer often go to bone or colon cancer to the liver?
It comes back to that seed and soil idea we touched on. First, the seeds the cancer cells have to actually reach the organ. Anatomical factors like blood flow patterns play a role. Colorectal cancer cells drain directly to the liver via the portal vein, making it a common first stop.
Okay, so plumbing matters, but what about the soil?
The organ environment itself has to be compatible, or the cancer cells need to adapt to it. Maybe some disseminated cells already have traits that make them better suited for, say, the bone environment versus the lung. That's selection.
Or they adapt once they get there. Plasticity again.
Exactly. Plasticity allows them to adjust. We see examples like breast cancer subtypes having different gene expression patterns that correlate with spread to brain, bone, or lung. Once in the brain, they might change their metabolism to use the specific nutrients available there.
And the cancer cells can change the organ environment too, right? It's a two-way street.
Definitely. They interact and co-evolve. Metastases in one site, like bone, can even release factors that promote further spread to other organs, like the lungs or liver. It's a dynamic interaction.
It really emphasizes that metastasis isn't just about the cancer cell itself, but this whole ecosystem. The review also talks about reprogramming the systemic macroenvironment beyond just the local TME.
Right. Metastatic cancer isn't just a tumor here and a tumor there. It affects the whole body. Systemic factors, nutrition, metabolism, hormones, inflammation levels can influence every stage of metastasis. And researchers are really starting to explore this inter-organ communication.
We talked about pre-metastatic niches. What else influences things body-wide?
Well, conditions like obesity or chronic inflammation associated with aging seem to promote metastasis. Conversely, things like exercise might reduce risk. Even our gut microbes, the microbiota, are getting attention.
The gut microbiome affects cancer spread. How?
Their metabolic products can get into the bloodstream and influence tumor cells or the immune system. Some bacteria might even travel to tumors and directly affect the microenvironment. There's also the whole emerging field of cancer neuroscience, how nerves interact with tumors. And even our daily circadian rhythms seem to impact things like when tumor cells disseminate or how effective our immune response is.
It's just layers upon layers of complexity. And then there's cachexia, that wasting syndrome.
Yes, cachexia is a really devastating systemic effect, much more common when cancer has metastasized. It's this profound loss of muscle and fat, extreme fatigue. It weakens patients, makes it harder for them to tolerate treatment, suppresses immunity, and is strongly linked to mortality.
Which brings us squarely to therapy resistance. It sounds like the very traits that make cells good at metastasizing also make them hard to kill.
That's often the case, unfortunately. Adaptability, stress resistance, regenerative capacity, these can all contribute to therapy resistance. And that's why established macrometastatic disease is still so difficult to cure. The treatments themselves also exert selective pressure, favoring the survival of resistant cells.
So we have systemic therapies, chemo, targeted drugs, immunotherapy. They've made progress, but resistance is the big wall. The review mentioned different strategies for different stages, like targeting dormant cells.
Yes. Trying to block the initial spread hasn't worked well in trials so far, possibly because spread can happen so early. So a big focus now is, can we target those dormant micrometastases? Because they might have unique vulnerabilities compared to actively growing cells. The challenge is identifying which patients have these dormant cells and would benefit from a specific therapy, especially since most trials focus on advanced, visible disease. We need better biomarkers, maybe ctDNA in the blood, or detecting DTCs in bone marrow.
What about oligometastatic disease, limited spread?
That's seen as a potential window of opportunity. If cancer has only spread to one or a few spots, aggressive local treatments like surgery or focused radiation, often combined with systemic therapy, can sometimes lead to long-term control or even cure for some patients. The difficulty is knowing if it's truly limited or if there are other hidden micrometastases already present. Better biomarkers are needed there too.
And the toughest nut to crack, macrometastasis, widespread disease. Plasticity seems like the major enemy here.
It really is. The cell's ability to change makes it hard for targeted therapies to keep working. Targeting those fundamental developmental pathways they hijack seems logical, but it's tricky because those pathways are also used by normal cells, leading to side effects. People are looking at specific metabolic weaknesses in metastases or ways to target the supportive microenvironment. But honestly, we need more basic research to find truly unique vulnerabilities in these advanced, adaptable cells.
It definitely feels like a huge mountain to climb, but the research direction seems clear. The review also had some intriguing final thoughts, like maybe metastasis isn't always a late event, that it could be separate from just tumor growth.
Yeah, that's a potentially paradigm-shifting idea,
Yeah.
challenging the dogma that you get a big primary tumor, then it spreads. Animal models show that cells, even precancerous ones, can sometimes disseminate very early.
What does that imply?
It implies that just targeting the things that make the primary tumor grow might not be enough to stop metastasis.
Yeah.
We might need strategies that specifically target the ability to spread, maybe much earlier, potentially even as part of prevention for high-risk individuals.
And what about the new tools helping us figure all this out?
There's been an explosion in technology. Single-cell sequencing, spatial transcriptomics, letting us see exactly what individual cells are doing and where. Powerful computational tools to analyze all this data. We can track cell lineages using DNA editing, analyze tiny amounts of material from liquid biopsies. The challenge is making sense of all the data and translating it into effective treatments, which requires better models, organoids, humanized mice, and maybe even using AI to help design smarter clinical trials.
This has been incredibly insightful. So to try and wrap this up, metastasis isn't just cells moving. It's this incredibly complex, multi-step biological process. It involves genetic changes, yes, but also this amazing, or perhaps terrifying, cellular plasticity, hijacking normal body programs, all happening within a constantly changing microenvironment that the cancer itself influences.
Exactly. And layered on top of that are immune evasion and the ability to enter dormancy, which makes it even harder to track and treat and ultimately contributes to why metastatic cancer is so often resistant to therapy.
But as you said, the research is moving fast. These new technologies, new ways of thinking, like seeing metastasis as potentially separable from primary growth, focusing on dormancy, understanding the systemic effects, it does offer real hope.
Absolutely. It's a huge challenge, no doubt. But the pace of discovery is accelerating. Understanding these fundamental mechanisms, plasticity, dormancy, immune interaction, the TME is paving the way for smarter strategies, whether it's finding ways to wake up or kill dormant cells, making the immune system more effective against metastases, or targeting those co-opted developmental pathways more specifically. There's definitely reason for optimism.
Okay, so here's a final thought for you, the listener, to take away. If the research is suggesting that even apparently normal or precancerous cells might have the ability to spread under certain conditions, what does that really mean for how we think about cancer risk, and maybe even how we approach early detection in the future? Something to ponder. We really encourage you to look into the resources we discussed. Thanks for joining us on the deep dive.